To find the exact atomic arrangement of a protein, crystallographers use X-ray diffraction to generate an electron density map. But constructing this three-dimensional map requires phase data that is physically lost during the experiment. If you start with flawed assumptions to guess those phases, the math will weaponize those errors against you. This modern crystallographic refinement pipeline is a continuous, dual-space loop designed to force a flawed initial model toward the true structure. We enter this loop using molecular replacement, repositioning a chemically similar known structure into the unit cell to guess missing phase angles. During the Fourier reconstruction of an electron density map, phase data completely dominates amplitude data. Altering a wave's phase shifts its entire structure far more drastically than simply changing proportions. Because our search model is an approximation, it is inherently incomplete and contains coordinate errors. This means the phases derived from it actively misrepresent the reality of the crystal. This chart tracks a molecular replacement starting model, plotting R-value, phase error, and figure of merit across increasing resolutions. Focus on the red line for phase error. Above a 60-degree error, features become untraceable. Beyond 4 angstroms, starting phases approach random noise. Because phase dominates the math, an electron density map built strictly from an uncorrected search model will reconstruct the incorrect model. It hallucinates its own inputs, completely masking the true physical structure of the protein. If you look at an initial, phase-biased map, the contrast is poor and the layout is deceptive. The false electron density perfectly wraps around the incorrect coordinates of the starting model. It validates the flawed wireframe, while the true structural shadow remains faintly ignored in the background. You cannot fix this by feeding the biased model straight into a purely automated mathematical refinement algorithm. Large positional errors, like an entire chemical loop facing the wrong direction, exist far outside the convergence radius of reciprocal space algorithms. The computer only makes micro-adjustments. It cannot entirely detach a sidechain and flip it across a void. Escaping this trap requires an alternating system. A human must manually intervene in real space, using 3D graphics to rebuild the model, followed by the machine performing mathematical optimization in reciprocal space. But before the human can rebuild anything, the map itself must be cleaned. Without a statistical intervention to break the phase bias, the structural model will permanently stall in a hallucinated reality, unable to converge on the truth. To construct a clean map, crystallographers replace basic raw structure factors with maximum likelihood target functions. This is the equation for maximum likelihood map coefficients. To account for the exact errors in our model, it introduces a statistical variance parameter known as sigma A. Sigma A expands into 2 core components. The first is the Luzzati D factor, which specifically scales down the calculated structure factor based on coordinate positional error. The second component is the completeness ratio. This explicitly accounts for missing atoms in the partial model, shrinking the output further. Applying these terms effectively downweights the high-resolution data that we cannot yet trust, filtering out the noise generated by our bad phase estimates. Simultaneously, the maximum likelihood calculation explicitly models the difference between observed data and calculated data, actively amplifying missing true features. The physical result is a bias-minimized map. The false hallucinations of the starting model fade, and the true structure emerges for the human to trace. With a clean map established, the crystallographer steps in. They use real space graphics to manually trace the amino acid sequence into the new density. This human-led rebuilding fixes the large topological errors, but it inevitably introduces countless microscopic geometric flaws, like stretched bond lengths and implausible torsion angles. To correct these, we must shift back to reciprocal space for global mathematical refinement. Global reciprocal space refinement is the mathematical optimization of every variable in the model, the exact XYZ coordinates and B factors, against raw experimental amplitudes. The primary danger of reciprocal space algorithms is entrapment in local minima, as illustrated in this target function plot. Look at the red dot. The atom of a misplaced phenylalanine ring has fallen into the electron density of a water molecule. The global algorithm cannot temporarily worsen its score to climb the energy barrier, so it remains stuck in this high valley. This entrapment is exactly why the human must continually cycle back to real space to manually pull the coordinates out of these mathematical dead ends. Even if the model escapes local minima, reciprocal space refinement faces a second major threat, the data-to-parameter ratio, which leads to mathematical overfitting. We can understand this using a basic physics analogy. This graph plots data points recording the trajectory of an object dropping off a cliff, fitted with linear, quadratic, and 9-parameter polynomial functions. The 2-parameter red line is a poor fit, but the 3-parameter green quadratic line accurately describes the actual physical acceleration of a falling object. Now look at the 9-parameter magenta curve. It touches every single data point, dropping the mathematical residual, or R value, to exactly 0. But the object takes upward turns. The physics of the model have become impossible. This maps directly onto protein structures. A crystallographer can artificially lower their global crystallographic residual, called R work, by adding split conformations everywhere, or modeling background noise as discrete water molecules. Minimizing the global linear residual creates a dangerous illusion of fit. Math alone cannot distinguish between capturing true structural physics and simply memorizing random noise. To prove that a heavily parameterized model actually describes physical reality, crystallographers rely on cross-validation. This diagram illustrates how data is physically separated before the refinement process even begins. Focus on the isolated circles at the bottom. A random 5% of reflections, roughly 1,000 data points, are permanently sequestered away into a free test set. The computer is only permitted to optimize the model parameters against the primary working set of data. The model's true accuracy is then exclusively evaluated by how well it predicts the amplitudes of the sequestered free set. This enforces Occam's razor. If adding a complex parameter lowers R work, but the R free value stagnates, the parameter lacks predictive power and must be deleted. R free neutralizes the threat of overfitting by acting as an uncompromising judge, forcing crystallographers to mathematically justify every single atom added to the model. We track the entire life cycle of a protein structure on the scatter plot, charting residuals over successive refinement iterations. On the left, the model genuinely improves as the red squares representing R work and blue stars representing R free drop in tandem. The critical inflection point occurs on the right. Notice the widening vertical gap where R work continues to drop, but R free rises. This is the exact moment over-parameterization begins. The mathematically optimal, truest model exists just before this divergence, at the minimum negative log-free likelihood. Returning to the refinement pipeline. This is the engine of discovery. Real space maximum likelihood mapping to break bias, reciprocal space parameterization to fix geometry, and cross-validation to ensure truth, repeated iteratively until convergence. The process ends with a crystallographic velocity established by George Sheldrick. A macromolecular refinement against high-resolution data is never finished, only abandoned. A solved protein structure is never a perfect reflection of nature. It is simply the exact boundary where our mathematics can no longer extract any more physical truth from the raw data.